I'm Don Berry. I'm a statistician for Berry Consultants, and I'm interested in designing clinical trials and incorporating disease modeling within the context of clinical trials. One of the clinical trials that I've designed and I'm a co-principal investigator for is called ISPY-2. A co-PI is Laura Esserman, who is a breast cancer surgeon at the University of California, San Francisco. And this has led to sort of a whole different attitude toward drug development, to therapeutic evaluation that I call platform trials. And I'll tell you what platform trials are. In 2006, Janet Woodcock, who was and is the director of the Center for Drugs at the FDA, suggested that the improved utilization of adaptive and Bayesian methods could help resolve the low success rate and expense of phase 3 clinical trials. And ISPY-2 is in that context. 2013, she further suggested that the FDA has to turn the clinical trial paradigm on its head in the modern era of personalized drug therapies and helping them to get to the drug to market faster. In 2011, the FDA published a document advertising their innovations in drug development and biomedical innovation initiatives to improve products for patients. And they advertised the two things associated with drug development, and one of them was essentially ISPY-2 and the approach of ISPY-2, looking at a different way of treating breast cancer. So in the old days, and increasingly, or still today, the standard approach for a woman who has been diagnosed with breast cancer is to remove the tumor and then deliver therapy, hormonal therapy, chemotherapy, and of course today, biological therapies are becoming much more common. What a neoadjuvant approach is you leave the tumor in the breast and deliver the same therapies for a period of months. In ISPY-2, it's about 6 months. You deliver therapies, and then you do surgery. You remove the tumor, or where the tumor once was, and the end point is called a pathologic complete response, when the pathologist can't find any tumor, either in the breast or in the lymph nodes. And so the FDA indicates that they are, this is 2011, that they are going to have a guidance, which they've since issued, for using pathologic complete response, I call it PCR, for accelerated approval in high-risk breast cancer. And they further suggest that the multi-drug screening trial, such as ISPY-2, could lead to a pathway for accelerated approval, and this would speed the availability of targeted therapies for breast cancer. A word more about the neoadjuvant approach. We get the information pretty quickly in 6 months about, was the drug effective in this patient, depending on that patient's characteristics. And that is so different from the standard adjuvant approach, where when you remove the tumor, you have to wait to see if the tumor comes back. And the good news is that in most breast cancer, it doesn't come back. But that means that in order to evaluate therapies, we have to have huge clinical trials. And in the adjuvant setting, we've had 3, 5, 10,000 patient trials in order to evaluate therapies. And that can break the bank. not only the bank of the pharmaceutical company, but increasingly we're paying more and more for cancer therapies, and we can't afford it. And we can't afford to run these huge trials. This is part of that same document suggesting that in this design, we look at biomarkers and we try to identify which treatments are benefiting which subset of patients. What is a platform trial? This is my one picture definition of a platform trial. It's when different therapies from different companies usually from different companies, are included in the same trial and evaluated on the same basis against the same control, and to try to understand which of these drugs benefit which subsets of patients. This is an article in the journal Nature Medicine a year or so ago that addressed ISPY-2 and its concept and its application to other diseases. And in particular, it highlights an institute, an Innovative Medicines Institute initiative that is anteing up 53 million euros to fund partially fund an ISPY-2-like trial in Alzheimer's disease, and this is in Europe, and they have funded it, and Berry Consults is part of the consortium that's working on this and leading in the development of the design of the trial. And as it indicates, drug companies are lining up to participate, to have their therapies evaluated, and potentially combinations of therapies, combinations of experimental therapies. I like this photo. It says drug companies are learning to run trials in many disease areas with each other across the divide. It actually turns out that across the divide of drug companies is much more easy to make than it is across the divide of countries and continents, but that's another story. Platform trials that are now running or under development in many cancers, and I'll mention more about the cancers in Alzheimer's, which I just said, in Ebola, antibiotics, community-acquired pneumonia, scleroderma, and other diseases. So it's becoming, and I think it's going to change the way we do things in the future for the efficiencies associated with some of the things I'm going to be talking about. So ISPY-2 is a prototype. This is a Wall Street Journal article when ISPY-2 first got started. What was the concept? And so it spells out that this uses genetic profiles to highlight biomarker differences among patients to match drugs to patients with biomarkers that predict a benefit. And the goal is not the huge phase 3 trial, but a small focused phase 3 trial focused within biomarker subsets that have been identified in the context of ISPY-2 as being the right signature for this drug, that is where you should be developing the drug. This is a cartoon. I'll take you through a cartoon of ISPY-2, but I first want to show you the status quo, or what has been the status quo in cancer generally and in metastatic breast cancer in particular, where you have a population of patients. The top suggestion is you just do a one-arm trial to look at tumor response or tumor shrinkage, and then carry that into later a phase 2 trial in metastatic disease. And then, if it's successful there, consider it for the adjuvant setting or for the early breast cancer setting. The other possibility at the bottom, and it's become increasingly used in drug development, is to randomize to a standard therapy and to look at another outcome, not necessarily tumor shrinkage, but tumor progression. But the important thing that I want you to focus on this slide is the population of patients. It's homogeneous. It's just this dark blue representing no variability in the patient population. Of course, we know not only in breast cancer, but in every cancer and every disease, patient populations are heterogeneous. And we ought to consider that very specifically in the design evaluation of clinical trials. So ISPY 2. You see the population of patients is now this mixture of these various symbols. We adaptively randomize within the subsets of the disease. So if you're in a subset of the disease that is responding very well to a particular therapy, that therapy gets higher probability. And if it's responding sufficiently poorly, that therapy gets lower probability of being assigned to the patient. It's possible, and indeed I'll give you an example, it's possible that in some subsets you've zeroed out the therapy, and in other subsets it graduates with a success and is now in phase 3. And you can't do that in a standard type of trial. So I've shown this with 5 experimental arms. I'll say more about what in the actual trial we have. 5 experimental arms in standard therapy. Every one of the experimental arms is compared to standard therapy within these patient subsets. And as we go, let's say arm 2 graduates. Has shown its muster in a particular subset called a signature of the disease and is graduated into a phase 3 trial. As we go, we're no longer randomizing to arm 2. Perhaps arm 3 drops for futility. It's shown itself as not to be beneficial in any one of the subsets. Arm 5 might also graduate into perhaps a different signature into a phase 3 trial. And as we go, you see there's now holes in the possible assignments, and we add arms. So this is a process where arms are graduated, drop for futility, perhaps reach the cap in terms of sample size, and other arms are entered. You can even, and this is sort of neat, you can even nest within the trial a factorial design. So in this case, arm C, arm D, arm C plus D, and of course the standard represents a factorial design where as you go, you may learn that the single agents aren't doing very well, but at least we get some evidence about them. But on the other hand, the combination is doing very well, maybe in one subset of the disease. And so the arm C and D alone would drop, at least within that subset, and the combination continued. The goal, and indeed what drives the entire design of the trial, is looking forward, is looking into phase 3. And the criterion for graduating is that we've identified a subset of the disease that has at least an 85% chance of being successful against standard in a future trial, in a phase 3 trial, the focused phase 3 trial. So the process looks like this. We start out in the upper left. We begin the trial with equal randomization probabilities, and in the actual trial, we started with just 2 experimental arms. We calculate the probability of success for each signature. As we go, we evaluate whether the arm graduates, and if it does, we stop accrual to that arm, or stops for futility. And if it does, we stop accrual to that arm. And if it doesn't, either of those, then we revise the randomization probabilities depending on the data that's accruing. And we do this continually, and continue the trial, again, calculate the success probabilities, decide graduation, etc. So it's a circle. We keep going through this circle. And if accrual is permitted, is permitting, then we add experimental arms as we go. So just, I don't want to get too much into the details, and you can not pay too much attention to the actual markers here, unless you're interested in breast cancer. But this serves as kind of a template for other kinds of trials. Maybe the markers are genomic, as in this example, but maybe they're phenotypic. Maybe it's age, maybe it's severity of disease. And so in I-SPY 2, we have 3 markers. One is HER2. Another is hormone receptor status. That's what the HR stands for. And the third is mammaprint, which is a 70-gene prognostic indicator of risk of the disease. And so there are 2 by 2 by 2 or 8 cells that you fall into. Now, with 8 cells, there are a total of 255 possible signatures, which is huge. And some of them are completely uninteresting, or some of them have very few patients within them. So what we do is we restrict to 10 signatures that are biologically interesting and that are marketable. And I'm going to go through quickly what those 10 signatures are. One of them is all of the disease. Another is just the hormone receptor positive, hormone receptor negative, HER2 positive, HER2 negative, mammaprint positive, and then some subsets. This is particularly interesting. This is called triple negative disease. The hormone receptor negative is both estrogen receptor negative and progesterone receptor negative. And so this is triple negative disease. This is hormone receptor negative and HER2 positive. I'll come back to both of these last 2 examples in a minute. Still another, that's 10. There have been 2 drugs that have graduated from I-SPY 2. One is veliparib, it's a PARP inhibitor in combination with carboplatin, and it graduated in this triple negative setting. The other is neratinib, which is a HER family inhibitor, and it graduated in HER2 positive, not too surprisingly, hormone receptor negative disease. So in this process, it's, as I indicated, neoadjuvant breast cancer. Primary endpoint is PCR. PCR takes place at the time, the assessment of it takes place at the time of surgery, which is about 6 months after randomization. But we get MRIs over the course of time. We get 2 intermediate MRIs, and we use them to address the question, so what is happening to this woman's tumor? And maybe it is suggesting that the tumor is going to be gone at surgery. That is, that path-CR, pathologic complete response, is likely at surgery, in which case we incorporate that information in the appropriate way, that is, with the appropriate uncertainty in the prediction. So MRI is not itself an endpoint, but it's a helpful factor that informs PCR. This is a never-ending screening process. It was sponsored by the Foundation for the NIH, which is a consortium of the Biomarker Steering Committee of the FNIH, the NCI, National Cancer Institute, the FDA, which has been instrumental in developing I-SPY 2, and industry. It's coordinated with the various divisions and the various centers in FDA, which, as I indicated, have established a regulatory pathway for neoadjuvant breast cancer. We have 20 sites. This is in 2014. We have 20 sites, over 700 patients randomized. The first 8 drugs were these 2 that I've mentioned, but we also have 2 Amgen compounds, a Merck compound, 2 Genentech compounds, and a Synta compound. These are the 2 that graduated. The first one, veliparib and carboplatin, was presented 2013, December, at the San Antonio conference. And this is the result in the triple negative signature. Veliparib carbo had a 52% PCR rate with that distribution shown in the red. And the control for triple negative disease was 26%. So there's a doubling of the PCR rate. And the probability of success in comparing veliparib carbo to control in a phase 3 trial, a modest size phase 3 trial of 300 patients in total, is 90%. On the other hand, this is the HER2 negative hormone receptor positive as opposed to hormone receptor negative, which you just saw. And in this subset, the control is actually doing numerically better. Not statistically better, but numerically better. And so the appropriate signature clearly within these factors is the triple negative disease. Neratinib was presented in April 2014. And as you see here, these are the two histograms showing neratinib with about a 56% PCR rate against control of 33% PCR rate in this signature, the hormone receptor negative, HER2 positive, with a predictive probability of success in a future trial of 79%. Neratinib was head-to-head against trastuzumab, Herceptin, on top of taxane, paclitaxel, in the first 12 weeks, as opposed to other therapies which are being added to the standard therapy of trastuzumab plus taxane. Interestingly, and the reason I show this is because of the 2 zeros there. Toward the end of the trial, in the latter part of the trial, it had learned, the trial had learned, the algorithm that was making the assignments had learned that the drug was not doing very well in this subset of the disease, and so it was assigning with probability 0. On the other hand, as I've just said, it graduated in the HER2 positive hormone receptor negative subset. So the idea is to match drugs with biomarker signature. There's, even before you do anything clever like adaptively randomize to make things go through faster if they're doing better in a subset, there's a savings from using a common control. So if you had, let's say, 5 drugs, and you're going to do 5 trials, 2-arm trials, randomizing equally, say, between the experimental arm and the control arm, that's a total of 10 arms. Whereas in something like a platform trial, there's 6 arms. There's the 5 experimental, and that against the common control. Better therapies move through faster, and the idea is this pairing of drugs and biomarker signatures to make for a more successful and faster and smaller phase 3 trial. One of the advantages, and it's a bit esoteric, but I want to point it out. Along the way in ISPY 2, pertuzumab was approved as for accelerated approval in HER2 positive breast cancer. And our investigators told us, and the DSMB told us, you've got to drop your standard arm because your standard arm doesn't have pertuzumab in it. And so what would happen in a typical trial if you had to drop your standard arm? It would stop. The trial could no longer be conducted. You'd end the trial and do the analysis and hope you get something that's valuable from it. In ISPY 2, we continued without a pause. We just dropped the arm in the HER2 positive setting. And we have these indirect comparisons across all of the arms so that we can still make comparisons against the original control, because we have these comparisons with all of the experimental arms to the control in the earlier time when the control was around. As we progress and continue with the trial, we'll be focusing on pertuzumab plus trastuzumab plus the taxane as being the comparison in the HER2 positive setting. So these are comparisons of modern platform trials in cancer. ISPY-2 is what I've been talking about. MyCat is a melanoma trial. It's melanoma. The MyCat acronym is Melanoma International Consortium for Adaptive Trials, and we're working on building it. It hasn't started as yet. BATTLE is a trial that if you're into cancer research you would have read about. It was a single institution trial at MD Anderson in lung cancer, 250 patients, looking at 5 different subsets, biomarker subsets. And the various things down the left-hand side tells you about the possible characteristics. Did the trial use combination therapies? Does the trial have the aspect that regimens come and go? Or do you just start with a particular set of regimens and then when you stop, you still have those same regimens in it? Does it try to understand what is the signature? And you see that ISPY-2 accomplishes most of these. LungMap is a highly touted trial that's being funded by industry, working with the National Cancer Institute, through the cooperative groups of the National Cancer Institute. It's really it's a master protocol in the sense that it has 5 biomarker subsets, but they don't use the same they use different controls in each. So it really is 5 trials that are pasted together. And they have some very efficient characteristics, such as seamless phase 2-3. UK Matrix is a very similar trial in lung cancer that obviously is in the UK. And GBM is glioblastoma multiform. This GBM Global is a trial that I'm helping the National Biomarker Development Alliance to set up in various countries, the United States, of course, and Europe, and Australia, and China, perhaps. So it's in development, and I put checks on every one of these because we've not clearly decided which ones we're going to do, but we're going to do as many as we can. I want to highlight these 2. So learn about off-target effects and adaptive randomization. When you have both of these, you accommodate something that heretofore has been a hindrance in understanding drugs. Roughly speaking, there have been 2 approaches. One is you give the drug to all patients, and then afterwards you look to see which subsets of patients benefited. There are inferential problems associated with doing that because there's so many possibilities. But you could find a signature that way and then run another trial to sort of confirm the signature. So one approach is treat all, identify the subset. The other approach is to assume you know the subset and just assign to it. You're sort of looking for a sweet spot. And when you see that, then you develop it. Herceptin is an example. It was focused on only HER2-positive patients. And now this is now 20 years later, we're trying to understand maybe it benefits some other patients. So there are negatives associated with both of those extremes. The learn off-target effects for a targeted therapy and adaptively randomizing within signatures means you can do what we did in neratinib, where if you find out that it has no off-target effect, you zero it out. You keep going in the portion of the patient population that seems to be benefiting. So you've compromised between these 2 extremes, and in my view, you get the best of both worlds. One other point here is the longitudinal modeling. Lung map does seamless phases 2 and 3. In phase 2, PFS is the endpoint. In phase 3, overall survival is the endpoint. From my perspective, you'd ought to be modeling the relationship between those. In lung map, they're not modeled in any kind of correlation. They are separately modeled, and you graduate, you go from phase 2 into phase 3 based on progression, and then there's a whole different endpoint, and you sort of hope that there's a relationship between. My preference would be to model that relationship. So I've talked about ISPY 2, I've talked about the other platform trials. I think it is the wave of the future. It's so efficient, not only in the phase 2 setting, also in the phase 3 setting, and in the comparative effectiveness research realm. And think about it as phase 4, where we have therapies out there in the world and we're not quite sure. We know they're effective overall, but we're not quite sure which patients benefit more, and should we be using combinations of these. Combinations, at least in cancer and Alzheimer's and many other diseases, is the answer, if there is one, if we can find it. I look forward to platform trials. They're cropping up all over the place, and they are the answer to the question. So thanks.